Good evening, everyone. Welcome to Pitch 90. We are really excited to have you all here at UD. My name is Holly Michael. I am the director of the Delaware Environmental Institute. I'm also faculty here at UD in the Department of Earth Sciences and the Department of Civil and Environmental Engineering. Tonight we welcome all of you who are watching, our presenters, our judges. Thank you so much to our judges who are taking the time to be here, and also friends of the environment. And I'd especially like to acknowledge the hard work of our DENIN environmental fellows, graduate students who organized the event this evening. DENIN fellows, would you all stand up? If you're here, some of them are outside. Yes, thank you. A round of applause for them. So they work as a team in organizing Pitch 90 and also other events for DENIN each year. This event, in this case, is every other year. And I also want to say thank you to our DENIN team, especially Jeanette Miller, who runs our DENIN Fellows Program, and also to Karen Rodalunas, who is here, and Sam Clem. Are they in the room? There they are, and Tracy McMullen. So thank you, everyone, for all of your efforts. So I'd like to share a story of a really special former graduate student here at UD. He completed his PhD several years ago now, and he's special because when he was a student, he came to us and said he has this idea for an event, and he called it Pitch 90, and in which students would showcase their research in a way that everyone can understand. So he offered to serve as Master of Ceremonies that first event, and it was a huge success, and we've been fortunate that he's been willing to come back every Pitch 90 since then and be our Master of Ceremonies. So Josh Lamont is now faculty at Brigham Young University in geological sciences, and his passion for research and teaching and communicating science are still lighting up lives. And so with that, please join me in welcoming Dr. Josh Lamont.
Thank you, Holly. That was very nice. It makes me blush to hear you say about, that I was special. But I'm grateful for DENIN, to the University of Delaware. Pitch 90 is happening tonight and has happened, the 7 times leading up to tonight because the folks at DENIN were willing to listen to an idea, right? And then willing to give us a little bit of money and let us do it. And I don't think I fully thought it through when I proposed it, because I wanted to win something, and I've never won anything except these trips back to Delaware. I'm grateful for each one of you for being here tonight also. I do want to take a moment and recognize our judges. And so tonight, each one of our contestants, they will give us their pitch for their exciting research. And really, it is exciting research. And over time, you think about where the trajectory of these folks who are speaking tonight will take them, and it will take them into leadership roles throughout this country and others. And so we have the opportunity to hear them and what's going on tonight. And the people who will decide who the best of the best are our judges. And so I'm going to go ahead and read your name. You don't have to stand up, but I think everyone at least wants to see your hand. And so we've got Michelle Chirpa, Sam Clem, Christopher Noman, Daria Nicotina, and Paula Cardenas Hernandez, Pablo Huck, Karen Rodalunas, Tracy McMullen, Eva Snell-Backmeister, Benjamin Bagozzi, Ryan Harrington, Helen Fischel, Amarantha Reddy Venapusa, and Chris Williams. And so thank you all. Let's give them a hand now, because these folks are going to be working hard to give our contestants their scores. So everyone, I think, is probably ready for me to be done talking. I am too. And we're going to do a quick activity though to warm up, to get you involved. And I'm gonna say too, for the contestants, you guys should do this too. So behind that black curtain, there are some contestants back there that are really excited about giving you their presentation. And it's simple, right? We're just gonna count. But what I want you to do first is I want everybody to stand up. There we go. And I want you to find a partner. So I want you to turn towards your partner, face your partner. Okay? We've done this at Pitch 90 before. So if you've done this before, hopefully, that's right. If there's an odd number, this still might work, but you can figure this out. All right, I'll give you a second. You've got a partner. Let me see. Oh, yep, they're doing it too. There you go. Okay, what I want you to do is I want you to count to 3, but I want you to alternate. So you and your partner will say 1, 2, 3. Okay? I want you to work with each other, count to 3, and I want you to see how quickly you can do it. Okay? So I know that you're going to be able to do this, but I want to see how quickly you can do it. So as you start, then ramp up your speed, and when eventually you'll make a mistake, which you will do, and we all make mistakes. Mistakes are good, right? We should fail and fail often. When you do make a mistake, I would like you to do a clown bow. What is a clown bow, you ask? So clowns bow like this. Huzzah! Okay? So when you fail, I want you to do that. I want you to say, huzzah! Okay, and I want you to do it, everyone with me right now, just to make sure you know how to do this. Okay, on the count of 3, we'll say 1, 2, 3, and then clown bow, okay? 1, 2, 3, huzzah! Yeah, oh, you're so good. Okay, all right, so I'll say start, and then you'll count to 3. Ready, start. All right, good job. Okay, so you're failing. That's great. Don't sit down yet. We're not done yet, guys. That was easy. That was the easy one. Okay, so we're gonna make this a little more complicated. So now you will stay with your partner. You will still count to 3, but in place of a 2, you will clap. Okay, so 1, clap, 3. 1, clap, 3. Okay, and again, anytime you make a mistake, huzzah! Okay, ready? Begin. And go fast. Okay, there's a lot more clown bowing happening. I love it. Okay, I have one more challenge. We're going to make this even more fun. And so now you'll go from 1 clap 3 to 1 clap stomp. Okay, so this is testing your rhythm here, folks. So 1 clap stomp, and again, with your partner, alternating, and see how quickly you can do it. And when you fail, you say, huzzah! All right, ready? Begin. Okay, great job, guys. Great job. Go ahead and give yourselves a round of applause. It feels good to feel a little silly, right? So we will we're lucky to hear a number of folks tonight. I'm going to come out in between each person, and I will introduce them. I will give you their name and tell you from which college they come, and then they will give you their presentation for 90 seconds. And if they decide to go longer than 90 seconds, I will gently remove them from the stage. And I just ask that you, as the crowd, that you stay engaged and that you refrain from clapping until after all of the presenters for this first half. Okay, that'll make things go a little smoother. If you feel the need to do something with your hands when they're done, you can do jazz hands. Okay? So that's, if it's like you can do a really loud jazz hands if you want to clap really loudly. Okay? So starting off tonight, our first presenter is Fernando Bustos from the College of Electrical and Computer Engineering. So Fernando, the time is yours.
Did you know that the price of solar modules decreased dramatically over the last few years? However, the number of solar projects postponed by utility companies increased. Why? Well, the intermittent nature of solar energy stresses the electric grid, becoming a barrier to the integration of more renewables. Batteries are the key to overcome that barrier. They can store that energy and deliver it when needed to reduce the price of electricity and integrate more renewables. Of course, one solar residential battery cannot make a difference. But think about it. What about 1,000 or 100,000 of them working together? They can act as a distributed green power plant by utilizing that green energy produced by solar panels. My research applied the power of machine learning to process the weather data and control the electric load patterns to control those distributed batteries plus photovoltaic systems and produce reliable power, integrating more renewable energies and giving you and me an active role in the energy transition. Combining the power of batteries and machine learning, the dream of a more sustainable and greener electric grid can become a reality. Thank you.
All right, thank you. Yeah, those were really loud jazz hands, guys. Next up, we have Shivam Chauhan, who's from the College of Civil and Environmental Engineering. Shivam, the time is yours.
Imagine if there was a highway system that not only connected thousands of miles of land, but also served as a sustainable solution to climate change. Highways connect our cities, but what if I told you there was an extraordinary substance that could be added to highways in order to drive us forward to a greener, more resilient future? Buckle up, because biochar can be added on the side of highways to mile by mile travel us to an environmentally conscious tomorrow. Biochar is a charcoal-like substance produced through pyrolysis, containing properties such as increased porosity and increased soil water retention, therefore boosting soil fertility. My research is on the implementation of biochar to roadway soils, right off the side of highways. The goal of my research is to examine microbial activity of biochar versus non-biochar amended soils and understand the impact that will have on greenhouse gas fluxes, such as carbon dioxide and methane. Over an extended period of time, using my own engineered gas flux chamber, I hope to be able to measure values of greenhouse gas fluxes and examine the impact biochar will have on global warming potential. My research on biochar will pave us a path forward where highways will not only connect our cities, but our future. Thank you.
All right, thank you, Shivam. Next up is Spencer Toth from the College of Biological Sciences. Spencer, the time is yours.
It's estimated that there are more viruses on Earth than stars in the universe. Bacteriophages are a type of virus that specifically infect bacteria and are known to be the most abundant biological entity on Earth, residing in areas like our oceans and our soils. I study a type of bacteriophage that infects Bradyrhizobium, which is a nitrogen-fixing bacteria that forms symbiotic nodules on the roots of leguminous plants like soybeans. Soybeans are actually one of the largest crop exports of Delaware and the United States as a whole. And in order for farmers to produce such high yields, they often utilize commercial fertilizers, which are unfortunately prone to leaching from the soil, entering into nearby waterways, causing eutrophication and toxic algae blooms, as well as contributing to the greenhouse effect due to increased emissions of nitrous oxide. However, studies inoculating soybean crops with Bradyrhizobium have actually shown to increase crop yields and demonstrate the potential to replace these synthetic fertilizers. My research is to get a better understanding of the bacteriophages which have an influence on these symbiotic relationships. Bacteriophages can influence their host range, the host populations, nutrient cycling, as well as bacterial evolution. To get a better understanding of these bacteriophages, I analyze their genome, their morphology, their host range, and their infection dynamics. Optimizing biological nitrogen fixation is a crucial step towards the push of utilizing naturally derived nitrogen and ensuring the health and safety of our aquatic ecosystems, as well as contributing to climate change mitigation efforts. Thank you.
All right, thank you very much, Spencer. Next up, from the Department of Political Science and International Relations, is Daria Blinova. Daria, the time is yours.
Thank you. In 1992, the United Nations Framework Convention on Climate Change was first adopted as the collective response to the adverse effect that humans produce on the Earth's climate. Since 1995, the annual meetings have been held in order to ensure that parties to the convention undertake active measures to address climate change problem. These meetings have been referred to as conferences of the parties, or shortly COPs, that include the delegates from the member nations and accredited actors such as civil society groups and private sector. The success of addressing the climate change problem is ultimately rests on the will of global leaders and accredited actors who participate in the negotiation process during these COPs. However, how much do we know about the individual attendance and participation of these actors during the climate change negotiation forums? According to the existing research, not that much. Nevertheless, understanding who represents the COP delegations is paramount for understanding whose interests are represented and why certain decisions are made. In our work, we strive to understand the drivers in nature behind the repeat attendance of individuals who represent countries and organizations. We build the data set from the annual COP's attendance rosters and extract such information such as title, gender, affiliation, and so on in order to understand what shapes the diversity across the turnover and to what extent representation during these COPs is equitable and inclusive. Thank you.
All right, thank you so much, Daria. Next up, from the Department of Geography and Spatial Sciences, is Siamak Malakpour Estelaki. Siamak, the time is yours.
We've all heard about water pollution. Agricultural non-point source pollution contributes primarily to deteriorated water quality in receiving water bodies worldwide, which can endanger the aquatic ecosystem's lives. The non-point source pollution is mainly caused by excessive application of fertilizer to farm fields. The extra fertilizer is then flushed away to the nearby streams. So there could be negative consequences, such as poor surface water quality. It can fuel toxic algae bloom, which is very harmful to ecosystems and human health. In Delaware State, for instance, most of the watersheds require reduction in non-point source pollution to meet water quality standards. On the other hand, best management practices such as trees, shrubs, or grasses planted next to waterways, including rivers and streams, can filter runoff, improve water quality, and provide wildlife habitat. Since non-point source pollution is mainly related to human agricultural activities, we need to tackle this problem from the perspective of coupled human and water systems. And my research tries to answer the question, how can incentives such as tax or subsidy impact on the farmer's decision to adopt agricultural best management practices? Thank you.
Thank you so much, Siamak. Next, from the Department of Human Ecology at Delaware State is Kelvin Ofori. Kelvin, the time is yours.
Have you ever eaten a food where a few days or hours later you experience symptoms of nausea, diarrhea, and stomach pain. These are symptoms of foodborne disease. According to the World Health Organization, foodborne disease causes over 600 million cases and 420,000 deaths annually. In US, one of the most common sources of foodborne disease is oysters. Oysters are filter feeders, meaning they filter through the aquatic environment. And as they do so, they tend to accumulate microbial contaminants from the environment into their bodies, which might pose risk of contamination to consumers. At Sally Cove in Rehoboth Bay, Delaware, is a principal habitat for these oysters, producing oysters for commercial benefit and for consumption. However, previous research reported that Rehoboth Bay, in addition to other Delaware inland bays, contain high levels of bacteria. Therefore, the objective of my study is to assess the presence of 10 pathogenic bacteria in these oysters. Preliminary results reveal that these oysters contain E. coli, Vibrio parahemolyticus, and Listeria monocytogenes in oysters that were sampled in July and August. Therefore, raw consumption of these oysters in summer might pose risk of contamination to consumers. Thank you.
Thank you so much, Kelvin. Next up, also from Delaware State at their Department of Agriculture and Natural Resources is Surya Krishna Saktivel. Surya, the time is yours.
Strawberry is one of the important food crops that's widely grown and consumed across the US. And it is a rich source of antioxidants and vitamins and other macro and micronutrients. Strawberry production, its quality and its shelf life are highly affected by fungal disease caused by Botrytis cinerea, which causes fruit rot disease in strawberries. So due to the repeated application of fungicides to control this disease, the fungus has evolved resistant to fungicide application. And this makes the situation more complex. It forces the growers to apply more fungicides than the optimum concentration to control the disease. The presence of fungicide residues on fruit and in the environment is highly toxic to humans and other life forms in the environment. So my study focused on identifying some of the candidate genes in the strawberry cultivar so that through various genomics and phenomics techniques and manipulate them using CRISPR-Cas9 genome editing technique to develop strawberry lines showing resistance to fruit rot diseases. So these fruit rot disease resistant cultivars will ensure the supply of high quality strawberries in the market without any fungicide residue, improved shelf life, and minimize the environmental pollution caused by excessive application of fungicide. Overall, it helps to sustain the strawberry production in an environmentally safe and sustainable way. Thank you.
Thank you, Surya. Next, from the Department of Geography and Spatial Sciences is Nazneen Nahar Sultana. Nazneen, time is yours.
Good evening. So welcome to the discussion and debates of climate change inequality and injustice. What makes inequality and injustice in climate policy and actions? Any guess? Well, there may be many factors and reasons, but one is very crucial. That is exclusion of the communities in the decision-making process from whom we are producing our policy actions. Let me explain. So consider yourself a community that is vulnerable, who lives in a coast. So the people who are living near in the coast in the front, they have different kinds of vulnerability from the back. There are also differences in socioeconomic and demographic profile. So as a single mom, I have different vulnerabilities than this handsome person. There are also differences in vulnerabilities who is 65 years old and who is 25. So considering all these issues, when we make policy actions from the very top-down processes, excluding all the specific needs of these communities, the outcomes may be inequitable. So I have taken questions for you. When we produce policy and actions, the question should be asked, what are the purpose of this policy? For whom? Who is participating in the decision-making process? And what kind of changes we are expecting from these policy actions at the very ground level. Thank you very much.
Thank you, Nazneen. That's the first time I was ever complimented in a pitch. Next up from the Department of Earth Sciences is Ophelia Christoph. Ophelia, the time is yours.
Are you willing to gamble with $1.1 billion? Last year, Bite Wind, an offshore wind company, bid $1.1 billion for an offshore wind energy area off the coast of New Jersey without knowing what sediments were underneath of it. My research looks at the sediments off the continental shelf on the coast of Delaware to reveal all of the sediments present in the area. The sediments in the area reveal a story of our past throughout time, through the different glacial periods, through sea level rise and fall, and these tell a story of our past to help us prevent damage in the future. These sediments tell us whether or not we need to prepare for engineering costs through the different foundation types, such as a monopile, a jacket structure, a gravity base, or a suction caisson, because each of these different foundations have different sediment requirements. And without considering these sediment requirements, the foundations could fall down. And we wouldn't want that. So we must consider these sediments early on in the process to save costs, so we can save billions of dollars in the long run, because we don't want to pay extra in legal fees either, because that would be bad. So in addition to the sediments present, we also should look at the hazards in the area so that we could prevent anybody from being damaged in the future.
Thank you, Ophelia. Next up from the Department of Geography and Spatial Sciences is Bokhtir Khan. Bokhtir, the time is yours.
Do you know which one is the most populous country in Africa? It is Nigeria. Currently, they have 200 million of people, and by 2050, the population will be doubled, which is 400 million. So imagine a place a little bit bigger than Texas, and with this huge number of population. Is that a problem? Oh yes, it's a problem if they don't have enough food to eat. So right now, like 7 out of 10 Nigerians don't have enough food for a day. So why they are not increasing their food production or crop production? It's not easy, because due to climate change, there's less rainfall and hotter temperature, which is a challenge for the farmers to grow the crop in the same piece of land. So what they are trying to do? The easy solution is like, let's cut down trees. They have a lot of trees and they are cutting down forest each year. Here comes my research. We're utilizing high-resolution satellite imageries along with machine learning algorithms to see how they're cutting down trees over 30 years and how this deforested land is getting converted into cropland, and the burning question is it really helping in achieving food security? So we have found around 42% of forest was lost in the last 30 years, and the fun fact is where there is less forest, more cropland, they are not getting much food. Rather, where there is more forest cover, they are getting more food. So what is the purpose of cutting down trees? So the farmers, they shouldn't do it. Rather, they should implement like irrigation practice or high-yielding varieties, which will increase the crop production and ultimately will help in food security. So this is the way that I can show, like, yeah, don't cut down trees, which is not good for the environment, and there's a win-win situation for both the environment and the people.
All right, thank you, Bokhtir. Next up from the Department of Chemical and Biomolecular Engineering is Alex Hansen. Alex, the time is yours.
Go to your spot. Lay down. 380 million metric tons. That's the combined weight of all plastic waste produced annually. To put that number in perspective, that's more than the combined weight of all humans alive today. The result of this problem is that every minute, a garbage truck of plastic finds its way into our natural waterways. I'm sure everyone has firsthand experience seeing plastic polluting our ecosystems and ruining their natural health. Imagine a future, though, where that plastic waste is able to decompose as naturally as the leaves that are on the ground right now. That may sound impossible, especially when I tell you that plastic in the ocean will take 400 years to biodegrade. Thankfully, recent research has revealed that yellow mealworms are capable of consuming and degrading plastics. My research focuses on understanding how the yellow mealworm and the microbes that live in its gut are capable of this plastic biodegradation so that we can then leverage this technology for other uses. Let's fast forward back to that future. Once my project is complete and we have an understanding of how the yellow mealworm and the microbes in its gut are capable of degrading plastics on rapid timescales, we could leverage that technology for producing medicines and valuable chemicals out of the plastic using those same microbes, or using those microbes for environmental remediation to degrade plastics at the source where they're polluting the environment. Thank you.
All right, thank you, Alex. Next, from the Department of Plant and Soil Sciences is Rachel Warthen. Rachel, the time is yours.
20 to 40% of our crops are lost annually due to pests and other diseases. Now, this number is rather concerning on its own. However, it's made more so by the fact that our projected population is 8.9 billion by the year 2050, which is a lot of hungry mouths to feed. Currently, some of our solutions involve chemical treatments in both fertilizers and pesticides that both help promote plant growth and prevent the spread of disease. However, the issue with this solution is that those chemicals can then get into our waterways, polluting not only our environment, but our food chain as well. The solution to this problem is right under our feet in the form of plant growth promoting rhizobacteria. These bacteria form special relationships with roots in which they not only help promote plant growth, but also help activate the plant's immune system in order to protect them from diseases. Through the usage of microscopy, I can look at how different small molecular weight compounds produced by the bacteria in the soil impact how and where these bacteria are attaching to the roots. Through analyzing this, I hope to help pave our way to a more food-secure future in which we use more biocontrols and less chemicals. Thank you.
Thank you very much, Rachel. Next, from the Department of Civil and Environmental Engineering is Narayan Kumar. Narayan, time is yours.
So if I ask you, what do you understand by the term turbulence? So you may think that there may be political instability somewhere, or there may be flight turbulence, or there may be a smoke coming out when you lit up a cigarette. That's true. When I study the turbulence, it's basically when the wave is coming down, impinging on the sandy beach, and that causing huge turbulence there. So why I need to study that turbulence? Because that turbulence is causing the sediment, erosion, and deposition. So you may ask that, do I need to track each sediment particle? No, that's not going to happen. I don't need to track each sediment particle. I use open form called sediment transport phenomena, and that is based on the open form technology. And I use that, and based on that, I used to calculate sediment transport. But why I need to study that sediment transport? Because you know that beach tourism and coastal infrastructure having a trillion or a million billion dollar of infrastructure. And when we will understand that, we will build either gray or nature-based solution to implement and protect the coastal erosion for the betterment of our society from this climate change. Thank you.
Thank you, Narayan. Next is Casey DeRoe from the the School of Marine Science and Policy. Casey, the time is yours.
So we all know that photosynthesis is a critical process to all life on Earth. Not only does it supply us with the oxygen that we need to breathe, it also supplies all organisms on Earth to some capacity with the high energy molecules that they need to power their essential life processes. But what if I told you that the photosynthesers that don't supply us with our oxygen are actually just as important to us as the ones that do? One such group of organisms are known as the green sulfur bacteria, and while they're not the best smelling microbes on the planet, their services to the environment and their potential to serve humanity make them an irreplaceable companion. They serve as primary producers in the deep sea sediments where it tends to be anoxic and low in light, where we won't find our typical photosynthesizers like plants or cyanobacteria. They can also completely detoxify sulfide into harmless sulfate in these environments, which makes them a little more comfortable for the few organisms that call it home. Additionally, they can produce wax esters, which are key precursors to biofuels, which is of interest to those who are searching for more and more ways to create these biofuels as we move forward into the future. This is just a few examples of the wide array of metabolic capacity that the chlorobi have to offer us. However, over 40% of their genetic material is still unknown to us. My research hopes to elucidate these unknown genotype to phenotype relationships, and by unlocking this deeper understanding of how the green sulfur bacteria work, we may also be able to find ways to make them work for us. Thank you.
All right, thank you so much, Casey. Well, that wraps up everyone for this first half, so now you can turn your jazz hands into a full clap, and let's give them a good round of applause. Really fascinating stuff, from worms that eat plastic to nutrient management and policy decisions. Really fascinating stuff. And one of the things that I love about what I'm hearing from this is just that their research is looking forward to solutions, right? And it's a wonderful thing. So now we can continue talking about what they've presented so far with an intermission. I think we've got about 15 minutes. There's some light hors d'oeuvres in the back, and so you all can go back there. After about 15 minutes, we'll come back into here. So thank you very much. Okay, welcome back, everybody. Hopefully you're feeling good and your brain has rested for a few minutes and you are ready to be wowed again at the wonderful research that's going on here. But now we've got a new round of contestants that are also really excited. I asked, maybe it was the first round, I said, how are you guys feeling? Are you excited? And they're like, well, that's not the word I would use. But I do want to take a moment and make sure we're still engaged. And so I'm going to ask everybody to stand up again. I know, yeah, don't get comfortable yet. The judges are getting their pens and papers out. You're gonna need your hands for this one, yep. And you guys, you know the drill, you're up and ready. Okay. So I would like you to, if you got anything in your glass, hurry up and drink it fast or find a safe place to put it. And what I want you to do is I want you to find a partner, but not someone next to you. I want you to look around the room and, it's like some enchanted evening and catch someone's eye. Okay. If no one's looking your way, then jump up and down somehow. You don't have to get close to them. You can stay where you are. Okay. And with a lot of science communication and communication in general, one of the things that is key to being understood and to understand is to have that back and forth between the speaker and the listener, right? And so when people who do this sort of thing professionally and get up in front of people, learn ways to engage with their audience. Lots of times we as science communicators do not learn those ways, and so we are left to our own devices. But one of the ways to engage with your audience is to make sure that you are seeing them and that you're taking their feedback. And if you need to adjust your message because it's not landing, then you do that. And when I teach my undergraduates at 3 p.m., lots of times I'm making adjustments. But right now we're gonna do an activity that practices that. And so I want you to get your partner, and one person is going to be the person who's throwing something. So decide who that is. Okay. Okay. So you've decided, and you're going to be the first person who throws it. The other person will catch it. And what I want you to do is I want you to act as realistically as you can, like you're tossing a ping pong ball to your partner. Okay? And your partner needs to catch it as if you would catch a ping pong ball. Okay? Go ahead and do that a few times. So the size, it needs to be size appropriate. Okay, very good. All right. Some of you look more athletic than others. Next up, I want you to act like you're throwing a bowling ball. Okay, so heft the bowling ball. This is like a 12-pounder. and throw it, and I want you to throw it in the air and catch it. That's right. Oh, gosh. Okay, very good. And lastly, I want you to do a beach ball to each other, okay? So go ahead and toss the beach ball. All right, very good. Go ahead and give yourselves a hand. You guys are wonderful ball tossers and catchers. This is also a great activity if you have boys that are, like I have 2 young boys, and they always want to throw or kick something in the house. And so, yeah, right. So that is, I like to do that activity because it teaches us and demonstrates how much what and where the message lands, like depends on what we're doing as the presenter, in addition to what you're doing as the audience. And so there are lots of friendly faces in the audience today, and I'm grateful for you guys for being here and supporting these folks that you will do now in the second half. So we'll go ahead and get started. From the School of Marine Science and Policy is Sarah Sweat. Sarah? She's not here. So that was a good practice. Next, from the Department of Entomology and Wildlife Ecology is Meredith Lewis. Meredith, the time is yours.
Owners of a building in Chicago found themselves in a bit of hot water on the morning of October 5th, 2023. Lights had been left on during a large migratory bird movement the previous night. That morning, nearly 1,000 birds were recovered after they collided with the building as they ventured south. Now, this number is staggering on its own, but for those of us who want to know more about the lives of these birds deeply, there was not just the question of how many collided, but of who collided. I scanned photographs that morning and noted some familiar faces: palm warblers, magnolia warblers, soras, and noticed the curious absence of others. Why is this the case? Is it route fidelity? Are some species more vulnerable than others? Or is it simply a matter of wrong place, wrong time? In bird migration research, we often estimate the numbers of birds flying high in the air without necessarily knowing a lot about their identities. My research uses automated recording units and new AI detection algorithms to listen to the community of birds flying high in the air to start to get at that question of who is up there and how do they respond to light pollution. I am also looking at how factors such as habitat and light levels affect how these birds aggregate together or when they stop over on the ground. With this information, perhaps we can more effectively plan efforts to turn off our lights, identify vulnerable species, and make those highways in the sky a little bit safer. Thank you.
All right, thank you very much, Meredith. You guys, your jazz hands are so good. Next up, from the Department of Chemical and Biomolecular Engineering is Austin Morgan. Austin, the time is yours.
In the 1993 film Jurassic Park, Jeff Goldblum's character had an iconic line: "Your scientists were so preoccupied with whether they could, they never stopped to think whether they should." Today, genetic engineers are making modified bacteria capable of fighting our pollution crisis by degrading plastic, and engineered fungi that can clean up freshwater resources by removing toxic contaminants. For these solutions to be most effective, they require the environmental release of engineered microbes. It's something that we could do, but is it something that we should do? Many scientists think it is, but acknowledge that we need safeguards against unintended consequences. A released microbe could be pathogenic for an animal important to its ecosystem, resulting in severe unintended ecological disturbances. Ideally, we would build off switches into the organisms that we release, allowing us to deactivate them once they've accomplished their purpose. But for an organism spreading in the environment, how do we find it to activate this off switch? Instead of looking for a needle in a haystack, we'd be looking for a needle in the Amazon rainforest. My research aims to solve this problem by developing a new tool for biocontainment called Time Bomb. Time Bomb is a programmable biological timer made of DNA and enzymes. We could put this timer inside of a genetically modified organism where it would silently tick, allowing the organism to spread and help us clean up our environment, before ultimately triggering an off switch in the form of a toxin that's produced to kill the organism. If my research is successful, it could enable the widespread yet safe deployment of engineered microbes capable of helping us solve our most difficult environmental challenges. Thank you.
All right, thank you very much, Austin. Next up from the Department of Biological Science is Krishna Thakkar. Krishna.
Okay. Good evening, everyone. As we entered the reception today, all of us enjoyed the warmth by the fire. We feel warmth of the fire from far away, but as it is close to us, we feel the burn. So today I'm going to talk about a thing which puts out fire, water. We have been using water bodies as dumping grounds for a long time. It's not a new thing, and we have been dumping plastic. When this plastic breaks down into small filaments, which are less than 5 millimeter, it is called as microplastic, which is not visible to our eye. But this microplastic gets lodged into the muscle of the very fishes that we eat. This plastic has been seen to cause neurotoxicity in such fishes. Now imagine the bioaccumulation and biomagnification happening in the food chain because of that. But this problem of water is actually on fire because it is closer to our home. In the hiking places like Boogie Run and Curtis Mill, our study, which happens every year with collection of water samples in the spring semester, sees how this microplastic content in the nearby water bodies has been increasing, which decreases the macroinvertebrates, chlorophyll A content, and algal species. So I can say that the water issue is actually on fire, and today when you go out, remember when you take the sip, are you drinking water or plastic? Thank you.
Thank you, Krishna. Next up from the Department of Plant and Soil Sciences is Kendall McCoach. Kendall, time is yours.
Forests store carbon, and this forest carbon storage is a widely adopted, government-endorsed, nature-based solution to climate change. But as cities expand, more forests are subjected to urban conditions. So how does urbanization impact forest carbon storage capacity? Fortunately, in this room today, we are surrounded by forests which have been embedded in our urban ecosystems for decades, sometimes even centuries. And the trees in these forests record history. Every year, trees produce a ring of new growth around their trunk. The width of this ring represents the tree's response to growing conditions like climate. Studying the variation in these rings throughout time tells the story of how this tree has responded to changes in climate and urbanization. By combining this analysis with tree inventories, I can produce detailed estimates of forest biomass, a proxy for carbon storage. This provides a much-needed understanding of our urban forests, which is so important because they comprise the majority of forested area across developed landscapes like the East Coast. Ultimately, we have decided to rely on forest carbon storage as a solution for climate change. So we need to understand the capacity of the forests that surround us to provide that service. Thank you.
All right, thank you, Kendall. Next, from the Department of Geography and Spatial Sciences is Manan Sarupriya. Manan, the time is yours.
Thank you. The east coast of the United States is experiencing sea level rise at a rate twice the global average, putting our coastal farmlands at a risk of a silent threat of saltwater intrusion. Imagine unwanted saltwater creeping into the very farmlands that feed us. It's like a domino effect that hits your plate directly, where climate change is also making it worse by introducing more severe and frequent storms, floods, droughts, which makes it all the more important to have a very sustainable management and carefully monitoring system for these farmlands. My research uses satellite data and machine learning to build a model which acts like a smartwatch for these farmlands. It monitors where is saltwater intrusion occurring, which is equivalent to a human health issue, and it also measures the progression of it in both space and time. It's like a constant check on the pulse of your food sources. Previously, it was very difficult to monitor saltwater intrusion because it's very tedious and it requires for the person to be on the field most of the time. We are solving this with our groundbreaking method that uses freely available satellite data that covers the entire Earth every 5 days, giving us high-resolution information without putting our boots on the ground. Our method is computationally very inexpensive that makes it usable for researchers from developing nations as well. We are securing what lands on your plate 1 pixel at a time. Thank you.
Thank you, Manan. Next, from the School of Marine Science is Tianyin Ouyang. Tianyin, time is yours.
Submarine groundwater, the water from continental aquifers into coastal ocean, impacts the surface water quality and geochemicals by transporting nutrients, organic carbon, and contaminants. It's also a large but understudied contributor to climate change and can release up to 13 teragrams of highly labeled organic carbon to the surface waters annually. This exported labeled organic carbon tends to be rapidly converted to CO2 in surface, contributing to 1.77 million metric tons CO2 in the US, which is greater than 1/3 of the 23 major sources reported by US EPA. Locally, in Delaware inland base, submarine groundwater accounts for around 80% of freshwater inputs and hypothesically to transport abundant nutrients and organic matters to the surface water bodies, which likely causes eutrophications, hypoxia, and toxicities in surface water. My research, therefore, studies the geochemical cycles of submarine groundwater in the inland base and will thoroughly answer how it impacts climate, water quality, and our coastal life in general. The outcome of my research will be compared with international data sets to develop a conceptual model that benefits the water source management and Clean Water Acts in the US and more broadly. Thank you.
Thank you, Tianyin. Next, from the Department of Entomology and Wildlife Ecology is Destiny Mann. Destiny, the time is yours.
Thank you. Huge problems face the world population today, such as poor waste management, expanding insect species that are invasive, and the struggle to provide a sustainable protein for the rapidly growing population. As an example, in 2018, 63 million tons of food waste were thrown out from US households alone. Of that 63 million, less than 1/3 was managed in any way, recycling or composting, and that left 40 million tons going directly into our landfills. With such huge problems, I think that if we want to manage and understand these problems better, we need to think a lot smaller. Edible insects. So in Europe, 4 species have already been approved, and that's the migratory locust, the house cricket, the yellow mealworm, and the lesser mealworm. The lesser mealworm is actually approved recently in this year and is a common poultry industry pest. If we're able to use these poultry and pest life history traits, like being able to reproduce them in very cramped spaces, in low nutrient available environments, and with a very quick life history, then we can actually reduce waste and produce a sustainable protein that can be harvested at the same time. So my research is optimizing this system. So I'm going to feed them a variety of different feed waste substrates, and I'm going to determine which waste is actually going to be reduced the most while producing the most protein during growth and development.
All right. Thank you, Destiny.
Thank you.
Next, from the Department of Materials Science and Engineering is Shai Santiparat. Shai, the time is yours.
Have you ever wondered what would it be like if you could use solar energy everywhere, not just from the shiny blue rooftop on your house? Let me introduce the flexibility to the world of the clean energy. There's a material called perovskite, low cost, less polluted, easy to make, and the most important thing, it can be made very thin. Looking at your hair, it's pretty thin, right? Perovskite can be made 100 times thinner than that. And that allows flexibility that can be mounted on any curved surface, such as on the car, or even on the fabric. That's impossible for the conventional solar cells. Now, if perovskite is so great, what are we waiting for? The problem with the scalability. Right now, the conventional method that we use to fabricate perovskite is for the liquid-based method. And that allows only a small size of solar cells. And if we increase the size, it's going to lose the uniformity very fast. And it's where my project comes to play. Now, I'm using the vapor process called vapor transport. So instead of using a liquid-based material, I'm using a gas-based material that helps spreading the layer more evenly. And no matter how large the material I make, it's not going to lose uniformity. And eventually, this method is not going to just stuck here in my lab. It's going to be a game changer to the world of renewable industry. Thank you.
All right, thank you, Shai. Next, from the Department of Plant and Soil Sciences is Jack Dietrich. Jack, the time is yours.
One of the biggest threats to biodiversity here on the East Coast enters our ecosystems in the smallest and most inconspicuous way. The seeds of many non-native plants disperse into native ecosystems, most often through plantings in our gardens and parks. Because cities have the highest concentrations of human activity, native ecosystems within these cities, such as urban forest, are the ground zero for many of these invasives to establish and spread. So if we can predict which invasive plants may spread their seeds into these urban forests, we can not only mitigate biodiversity loss, but also prevent the further spread of these non-native plants. To do this, we can look to see what seeds we find within the soils of urban forest, giving us a sneak peek into the potential future of these ecosystems. We did this by sampling soils from 25 urban forests between North Carolina and Massachusetts. We brought these soils to greenhouses where they were monitored for germination, and we identified and counted each seedling that emerged. About 1/3 of all of the seedlings that we saw were from non-native plants. And in some of our forest plots where we saw one invasive tree, over half of all of the seeds from these plots were from that single invasive species. So we hope our study can help us understand how invasives become established in forests, and in turn, how we can maintain biodiversity within our cities. Thank you.
All right, thank you, Jack. Next, from the Department of Chemical and Biomolecular Engineering is James Buchan. James, the time is yours.
If we stop fossil fuel production today, we're going to continue emitting CO2 from vital industries such as agriculture and construction to the tune of 1 billion tons per year. We will need to be able to offset these emissions to meet our climate goals. And one of the technologies considered is direct air capture. Unfortunately, this is an expensive technology at $1,000 per ton. If we think about that at scale, we're talking about a $1 trillion per year price tag, something larger than the US military budget. The US has recognized the needs for this and has started providing research funds for research such as my own to reduce that cost to $100 per ton. When we started research, we looked at the current state of affairs and saw that the energy costs of current technologies exceed that budget, and we knew that we needed to try something different. We found nickel hydroxide from AA batteries could be a solution, a technology that's been around for decades. With this in our electrochemical cells, we can produce hydroxide which reacts with CO2 in the atmosphere to make carbonate. This can be separated and concentrated and sequestered underground. In our labs today, we have been able to get our energy costs down to $50 per ton, meeting our goals and something that we're looking to improve and scale to meet the direct air capture needs. With this and many of the innovations discussed today, perhaps we'll meet our climate goals.
All right, thank you, James. Next, from the Department of Civil and Environmental Engineering is Harrison Fleetwood. Harrison, the time is yours.
California taxes, New Jersey drivers, vegans. What do they have in common? A less than perfect and somewhat deserved reputation. Fracking suffers from a similar issue of public perception. What if we could bring a win to one of oil and gas production's least popular practices? One byproduct of fracking is produced water, a very salty, briny liquid created as a fracturing fluid. When it accidentally spills, it kills plants and microbes and degrades soil quality. The current remediation strategy: excavate, landfill, and replace the contaminated soil, coming at a great cost to both the environment and the reputation and bottom line of the oil and natural gas producer, a lose-lose situation. My research explores a new remediation strategy that can flip this issue into a win-win. When produced water spills, water evaporates up through the soil, while salts precipitate under the subsurface. These trapped salts are what create the dead zone in the contaminated soil. My research seeks to use crystallization modifiers to disrupt and delay the precipitation of salts. More simply, this means we aim for the salt to form on the surface of the soil rather than beneath it, where it can be cleaned off. This in situ remediation technique preserves the original soil while forgoing the heavy machinery required to excavate and transport contaminated soils. Oil and water don't mix, but oil and natural gas production and cost-effective, environmentally sustainable remediation practices can. Thank you.
All right, thank you so much, Harrison. And thank you to all of the presenters from this half. That wraps up this half and the evening. Let's give everyone a big, loud round of applause. Fantastic talks. For everyone here, if you go ahead and scan this QR code, you get a vote too. So this will be the fan favorite. You scan that QR code and you'll have the opportunity to vote for your favorite. So we will now, the judges will finish wrapping things up, and then the DENIN fellows will tally up the scores. And while they're hard at work, trying to avoid any hanging chads, we will, most people don't get that, the kids don't get that, right? We will move out, and I hear that there actually are hors d'oeuvres now, yes? All right. So let's go ahead and give these guys and gals another round of applause. And go ahead, folks, and you can make your way out and have the hors d'oeuvres. Thank you again.